Hello, everyone, and welcome to today's webinar, Overcoming Sequence Artifacts to Fuel Clinically Relevant NGS Applications. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. I'd like to now welcome our speaker, Dr. Zain Jaffer, Senior Scientist at Watchmaker Genomics. Zain, you may now begin your presentation.
Thank you for the introduction, and thanks for everyone who's taking the time to listen in. Before we get started, I'd like to take a second to introduce Watchmaker Genomics and the team of experts bringing this presentation together. We're a life science company headquartered in Boulder, Colorado, with an R&D facility in Cape Town, South Africa. We specialize in the design, development, and production of tools and toolkits that modify nucleic acids and enable high-growth applications in genomics, molecular diagnostics, and personalized medicine. With experienced leadership, our talented team has made great use of the last 2.5 years since our founding, and we're excited to bring these high-performance products to the market. In recent decades, we have all bore witness to unprecedented advances in genomics technology and have seen academic, industrial, and clinical genomics markets evolve alongside. As costs decrease and throughput and resolution increase, sequencers are moving toward becoming a universal readout for biological information. Sequencing is now being applied not only to genomes or transcriptomes, but to understanding the dynamic epigenetic landscapes of such samples. In addition, various technologies are appearing that allow the readout of proteomic as well as pharmacologic information. With so many important biological signals converging on sequencing, an industry mission becomes ever more prescient. The goal is to make the data recovered from an instrument be a perfect representation of the information carried within the sample. Capturing a sample sequence information requires enzymatic manipulation at multiple points from purification of the sample to preparation on a flow cell. As a result, data quality can only be as good as the enzymatic reagents used to process each sample. High-quality reagents are necessary for the development of highly sensitive, specific, and robust sequencing-based assays. If you control the enzymes, you control the information. This is particularly relevant when considering the needs of clinical sequencing applications. Over the past decade, the availability of higher quality reagents has enabled the use of lower quality samples and reduced input amounts. Clinical applications continue to push the boundaries of existing NGS technology and require reagents specifically formulated to address unmet performance needs. Sensitivity, specificity, simple and scalable workflows, compatibility with a broad range of sample types, and vanishingly small input amounts are necessary performance characteristics of the next generation of enzymatic tools for genomics. Watchmaker Genomics is uniquely situated to address these needs. At the foundation of our organization lies deep domain expertise in enzyme manufacturing, enzyme engineering, and computational biology. Our ability to identify, engineer, and manufacture enzymes at high purity enables accurate assessment of enzyme function and the ability to ideate, prototype, and characterize their function during rapid and dynamic iterative cycles. Focusing on application-specific variant selection and reaction optimization ensures that we're building tools for a purpose rather than finding a purpose for the tools that we build. Our development strategy, in simplified terms, is to combine rigorous enzymology with strategic experimental design. Our goal is to harness the power of design of experiment style experimentation and couple it to NGS readouts to parallelize our testing and, by leveraging our enzymatic and computational expertise, maximize insight. We start by finding, evolving, or designing enzymes to fit an application need. These molecules and the reactions they govern are often multi-step, so in order to really understand how they function, we assay individual activities in addition to endpoint measurements. An example would be optimizing DNA polymerase activity. In order to understand variant performance, we independently monitor characteristics like substrate affinity, turnover rate, and specificity. That approach, complex with DOE-style buffer screening and highly multiplex NGS readouts, allows us to acquire multidimensional and nucleotide resolution insight into the critical levers for tuning performance in each reaction. This sort of deep characterization of a complex reaction space provides us with an understanding of the chemistry that allows us to be agile during development and rapidly customize based on users' needs. One of our first exercises in applying this strategy was focused on DNA library preparation. Sonication is the gold standard for unbiased, robust DNA fragmentation, Unfortunately, it requires a large upfront investment in equipment, and it's not easily scaled to large sample numbers and automation without increases in overall processing time. Generating libraries from low inputs with this method is especially difficult due to sample loss inherent to processing and generally low sample conversion rates of available library preparation chemistries. Related to some of these limitations, sonication-based library preparation workflows have trouble fitting nicely into the evolving paradigm of clinical sequencing needs that I described earlier. DNA library preparation chemistries based on enzymatic fragmentation are an attractive alternative that alleviate the monetary barrier to entry and bottleneck of sequential sample processing and automatability. However, existing enzymatic fragmentation-based chemistries are not without their own limitations. Particularly undesirable are increased bias relative to sonication and the introduction of sequencing artifacts that result from use of previous or existing products. Any new enzymatic fragmentation-based product needs to successfully pair the sequence accuracy and low bias of sonication-based library prep with the scalability and input compatibility of enzymatic fragmentation-based products. We saw an opportunity to leverage our domain expertise and industry experience to tackle this problem. We had specific performance goals in mind: to lower bias, minimize artifacts generated during library preparation, deliver high conversion efficiency, as well as maintain flexibility with respect to input requirements for either high-quality DNA or DNA extracted from fixed tissues. Through the remainder of this section, I'll show you some of the data we generated that supports the assertion that we've done a pretty good job accomplishing these goals. We ultimately developed the Watchmaker DNA Library Prep kit with fragmentation and the following key characteristics and workflow details. Our workflow incorporates an integrated fragmentation, end repair, and A-tailing step, followed by a same-tube high-efficiency ligation reaction. This reaction is powered by a ligation master mix built to improve liquid handling and ease of automatability. PCR-free workflows can be finished in an hour and a half of reaction time, while PCR-based workflows require up to an additional 30 minutes with an additional SPRI-B cleanup. Our kit supports DNA inputs from 500 nanograms to less than 1 nanogram with consistent conversion rates and highly tunable fragmentation for all input amounts achieved just by adjusting the time and/or temperature of incubation. As I mentioned before, our research and development strategy is focused on efficiently sampling a large reaction space and understanding the multi-step activities of enzymes we use. As a result, we generate large data sets that, ideally, report reaction optima as well as reaction failures at extremes. Our efforts optimizing for decreased GC coverage bias is a great example of this strategy with real utility for users. Increases in uniform coverage ultimately lead to increased sequence economy and better data. Shown here is normalized coverage data from a series of PCR-free libraries generated from human gDNA under different reaction conditions modulating enzyme variants, buffer composition, or the protocol. Each coverage plot reports normalized coverage on the y-axis as a function of GC content on the x-axis. Not only have we identified how to minimize bias for this sample type, we better understand how to manipulate bias in this reaction in either direction from ideal. This type of comprehensive characterization serves two purposes. One, it allows us to quickly iterate and customize product performance based on future user needs. Two, it allowed us to produce a base product with desirable bias characteristics relative to existing products. Shown here is a data set generated from either our DNA library prep kit and two competitor kits. Again, we're reporting normalized coverage data from samples generated from high-quality human gDNA. In this particular experiment, coverage plots were generated from 1 nanogram of input and prepared according to manufacturer's recommendations, including PCR. What you see here is data showing that, while each kit performs similarly through the bulk of the genome, some differences appear at GC extremes. Overall, our kit performs more consistently with respect to GC coverage bias than other related products. But what use is uniform coverage if a significant portion of that incoming data is not representative of the sample itself? The artifacts generated during enzymatic fragmentation can, at the very least, lead to lost data and increased cost, but at its worst, create a very real problem for clinical variant calling. Chimeric reads are generated as either authentic and likely clinically relevant in vivo gene fusion events, or during library preparation when two fragmented molecules distant in linear sequence but now broken apart and close in 3D space are fused together. Industry leaders have pointed to these types of artifacts creating problems for variant calling because it's sometimes difficult to determine which of the two causes is at play. In fact, it's been pointed out that the number of chimeras reported in libraries prepared by enzymatic fragmentation may be 10 times more than reported in similar samples prepared by sonication. Our kit virtually eliminates that excess relative to sonicated samples. In the data shown here, we have compared the percentage of reads reported as chimeras for either libraries prepared by sonication-based prep, our chemistry, or two competitor products. Under similar circumstances, our chemistry reports chimera levels approaching that of sonicated libraries, a significant improvement from other chemistries. Processing of fragments during library preparation often induces non-templated or low-fidelity polymerization of few bases onto the end of library molecules, inflating the overall number of mutations and decreasing overall assay sensitivity. This sort of noise presents especially large problems detecting low allele frequency mutations. In addition, groups recently have observed a more complicated problem with these types of end-localized artifacts. Last year, Gregory et al. Presented an analysis of the artifacts commonly generated at the ends of reads when using enzymatic fragmentation-based kits. They suggest that, with sufficient sequence complementarity, hairpins may form at the ends of reads during the end repair step that may be resolved into sequenceable molecules through the mechanism they propose at the left of this slide. Regardless of the actual mechanisms that drive this phenomenon, this sort of contaminating data ultimately manifests as the problem shown at the right. When compared to libraries generated from sonication in the blue overlay, enzymatic fragmentation-based libraries in red accumulate significantly higher called variants shown on the y-axis as a function of variant allele frequencies shown on the x-axis. Notice how enzymatically fragmented samples show a much larger distribution of variants below 10% variant allele frequency than the sonicated samples. Often, noisy sequences like these at the ends of reads can automatically be trimmed or soft-clipped during analysis, which may only create a problem for sequence economy. But every bit of increased stringency of filtering runs the risk of removing true positives. More concerning than the soft-clip rates are those reads that are not soft-clipped due to aligning to semi-palindromic sequences. These non-clipped reads result in inflated SNVs and indel rates at the ends of reads due to the similar, but not identical, sequence character of the molecules they're derived from. Therefore, improvements in chemistry can directly increase both sensitivity and specificity of variant calling. Members of our bioinformatics team generated a similar artifact detection tool as that used by Gregory et al. And used it to visualize the frequency of hairpin artifacts generated by our chemistry, two competitors, and compared them to samples processed by sonication. This data is reported as a rate in the form of artifacts per million fragments. Again, similar to sonication-based library prep methods, our chemistry generates libraries with very few of these artifacts, each of which represents a potential false variant call. I've spoken about the importance of decreasing bias and minimizing artifacts in the context of variant calls as a function of improved chemistry, but a similarly important and straightforward path to improve data quality is to capture a higher percent of the input to begin with. To that end, we executed a head-to-head comparison measuring the library yield and found that our kit generates overall higher yield than some competitors when preparing libraries from either high-quality DNA inputs or from formalin-compromised reference DNA. The graph shown at the left reports the concentration of libraries in nanomolar on the y-axis. Each sample was prepared in triplicate from 25 nanograms of either high-quality gDNA, moderately formalin-compromised, or severely formalin-compromised DNA samples purchased from Horizon Discovery. Yields are shown after 6 cycles of PCR. The electropherograms on the right illustrate consistent library sizing among variable quality samples prepared with our kit. Our chemistry offers the performance increases I just discussed, but not at the expense of highly desirable characteristics like insert size tunability and compatibility with a wide range of inputs. Tunable fragmentation is a key advantage of sonication-based library preparation, and so ideally any enzymatic solution should perform well in this respect. Unfortunately, the lack of tunable and reproducible fragmentation has been a pain point for many users of enzymatic fragmentation, even going so far as to have great difficulty making libraries with insert sizes at or greater than 500 base pairs. Shown to the left are traces of libraries prepared only with differing fragmentation times. Within this panel, we show that not only is insert size tunable from greater than 500 base pairs to less than 200 base pairs, fragmentation is consistent between duplicate reactions. Fragmentation is also consistent across input amounts with acceptable ranges spanning many orders of magnitude. The panel at the right shows overlaid traces of libraries prepared in parallel from 500 nanograms down to 0.1 nanogram of input DNA. In fact, libraries can be generated from inputs as low as 1 picogram of high-quality DNA. Using human gDNA as input, we were able to generate libraries from 1,000 picograms to 1 picogram without a change in library size. The libraries generated in this experiment are not only sized appropriately, shown on the image on the left, but the vast majority of reads align to the reference without significant adapter contamination, illustrated by the graph on the right. Overall, our kit delivers high performance at vanishingly low input masses and expands the potential application space for this product into single-cell territory. With such low sample inputs, PCR is required to generate sequencer-ready libraries. As a result, a robust, high-fidelity library amplification module is a necessary companion to our library preparation kit. As I mentioned before, errors in sequencing data can come from any point in the multi-step process from a cell to a sequencer, and many groups have expended the effort to quantify the extent and source of errors. One important source is the fidelity of library amplification polymerases. A report earlier this year in Genome Biology set out to quantify the error rates of library amplification products, as well as characterize error rates of individual instruments at different institutions. In the figure shown in the leftmost panel, they report real performance differences between the two high-fidelity polymerases, kappa Hi-Fi and NEB's Q5, with resulting error rates differing multifold between these products. Fidelity problems in library amplification can sometimes be mitigated through UMI methodologies and error correction algorithms. However, sufficiently complex libraries or libraries generated without uniform amplification may result in many UMI families lacking sufficient depth to make this correction possible. As a result, any reduction in polymerase errors is beneficial to the end user. Following a similar strategy to library prep, we optimized for the fidelity of our library amplification module. Now in the context of PCR, we strategically investigated multiple polymerase variants, buffer components, cycling conditions, and component concentrations with a specific focus on the fidelity of PCR amplification. Shown at the right are polymerase error rates parsed by specific type of base substitutions. Two important conclusions are to be drawn from this experiment. First, a subset of errors predominate during library amplification, particularly A to G and C to T mutations. Both may arise as a function of base misincorporation and DNA damage during thermal cycling. Second, it is possible to reduce these types of polymerase errors by the right combination of polymerase variants and reaction conditions. Using this and many other datasets, we developed the Equinox Library Amplification Master Mix. This product is a robust and high-fidelity library amplification solution with usage and performance similar to Kappa Biosystems Hi-Fi, but with marked increases in fidelity and workflow compatibility. The first performance characteristic I'll point to is overall library amplification fidelity, a direct result of the development strategy I presented on the previous slide. Shown below is a comparison of the polymerase error rates associated with each base substitution for both Equinox and Kappa Hi-Fi. Overall error rates are lower with Equinox, and a particular performance increase shows up with respect to A to G and C to T mutations that I discussed on the previous slide as well. Relying on high-fidelity amplification systems allows high-confidence calling even when read depths are too low to allow for error correction. In addition to exhibiting low error rates, a high-quality amplification mix must amplify uniformly. Employing the use of a low-input, high-amplification, UMI-based model system, we investigated the amplification characteristics of Equinox, Kappa Hi-Fi, and attacked DNA polymerase 2X master mix. In an ideal case, no DNA molecules and associated UMI families would be inefficiently or preferentially amplified. That result would be that after amplification, all UMI family sizes would be evenly and tightly distributed. This is illustrated in the pink ideal line on the graph shown. This analysis reports the cumulative fraction of molecules or UMI families on the y-axis versus the fold change from mean UMI family size shown on the x-axis. The more quickly a dataset reaches 1, the more uniform library amplification has been. Amplification with Equinox, in purple, leads to greater than 80% of UMI family depth, independent of insert GC content or length, fall within 2 to 3 folds of the mean UMI family size. Kappa Hi-Fi also performs well, as shown in the green trace. For reference, a poorly performing polymerase, Taq DNA polymerase in NEB's 2x Taq master mix, in the orange trace, results in only 50% of UMI families falling within that range. With a focus on automation, I'd like to highlight a workflow improvement for Equinox. One potential problem with automated sample prep for PCR is the stringency of hot start performance. High fidelity amplification mixes have 3' to 5' exonuclease activity necessary for polymerase proofreading. Unfortunately, if not properly controlled either by low reaction temperature or by the hot start capability of a mixture, extended incubations, for example, long setups for automated workflows, of amplification polymerases and primer can lead to nonspecific activation of that activity and affect primer quality. Any promiscuous decay of primer or template at 3' ends can lead to decreased specificity of amplification, especially for low concentration samples. The hot start capabilities of Equinox inhibit not only the polymerase function of the mix, but also its exonuclease activity. Shown in the graph here is normalized polymerase and exonuclease activity as measured by dNTP incorporation or dNMP release, respectively. Overall, our Equinox library amplification master mix is capable of highly specific, highly uniform, and high fidelity amplification of NGS libraries and is a great companion to our enzymatic fragmentation-based library prep kit. At the outset of our development efforts, we had the goal of addressing some key challenges in DNA library preparation. We wanted to support the market by developing products that maintain the data quality of sonic-aided library prep and the simplicity and scalability of enzymatic library prep methods. What I presented here is performance data generated either using our Watchmaker DNA Library Prep kit with fragmentation, powered by Equinox library amplification. We have a straightforward and scalable workflow with highly tunable fragmentation, compatibility with a huge range of DNA input amounts, low coverage bias, and dramatically reduced sequencing artifacts. These performance improvements bring us one step closer to realizing the industry's mission of making the data out a perfect representation of the sample coming in. At Watchmaker Genomics, we have a unique and powerful perspective on how to enable the evolving genomics market. Our vertically integrated development platform combines specifically engineered enzymes into highly optimized reaction systems and end-to-end workflow solutions. We have the power to purpose-build applications-driven tools for the clinical space from the ground up. We're taking this novel platform and the insights gained during development of our initial enzymes, reagents, and workflows, and we're applying these concepts to the next wave of Watchmaker-grade products. Keep an eye out for upcoming information on our engineered reverse transcriptases and RNA-seq library preparation development efforts. And with that, I'd like to thank you for your attention and open this up to questions.
Thank you, Zane, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, how does library prep efficiency help with artifacts?
Yeah, that's a great question. Thanks for starting that off. I think the answer to that really comes from two places. I think the first thing is that not all molecules that are captured during library prep are generated with artifacts in them. And so catching more of the molecules from an input that don't have any artifacts or just having a more accurate representation of the sample is good from that perspective. So you just have a greater chance of those molecules ending up getting sequenced. In addition, higher conversion rates allow you to decrease the number of or amount of PCR, number of cycles that you have to go through, and also mitigate some of the potential artifacts that generate from or that come from that as well.
Okay, thank you. Our next question is, are there differences in the way that FFPE and high quality inputs are handled?
Yeah, great question. So, of course, for anyone who works with DNA extracted from fixed tissues, the sample qualities coming out can be quite variable. What it really means for compromised or low quality versus high quality samples is that the workflow is going to end up being exactly the same in terms of handling and steps and all that sort of stuff. But what will likely have to happen is that the fragmentation time or parameters may be modified depending on the sample. And that's ultimately the only point, I think, where that'll need to be altered.
All right, thank you. Next question, what is the minimum input required for PCR-free libraries?
So that's a bit of a nuanced question, just to answer that a bit. I think that depends a lot based on the library size you're targeting, and as well as whether or not you're looking to multiplex or any sort of things like that. But generally speaking, if you're looking to sequence a single sample, we kind of recommend that if you're looking for short libraries or libraries on the shorter end, that starting with 20 to 30 nanograms is usually pretty safe. And if you're looking for very long libraries, something on the order of 40 to 50 nanograms. And of course, that's going to vary by some other things related to sample quality and all that sort of stuff. But we think that's a pretty good starting place.
All right, thank you. Next question. Can you comment on performance of fragmentation module with contaminants, such as EDTA or from crude extracts like blood, saliva, etc.?
Sure. This product, or the I guess library prep side, is formulated in such a way that it accepts either water, standard tris buffers, or buffers containing up to 0.1 millimolar EDTA in the total sample coming with 0.1 millimolar EDTA. Problems or protocol adjustments or anything like that. With respect to some contaminants, we have not done a lot of testing on those sorts of samples, or those sorts of crude samples, let's say. But preliminary experimentation shows that our product actually works pretty well with using cells as input, or a few cells as input, but we don't really know about the blood or saliva, but we do have plans to explore this a little bit better in the coming months as we look a little more into it.
All right, thank you. Next question. Does your polymerase extend across uracil and DNA?
It does not. We do have a product that would fit that need, and I think if you can check in with our sales team, then we can get that to you.
All right, great, thank you. Next question, is this product compatible with inputs in TE buffer?
It is. I think it depends on let's say the strength of the TE buffer related to that, what I mentioned earlier about EDTA. And anything coming in with 0.1x TE works perfectly well with. But again, depending on let's say the X of the TE, then there may be some fragmentation time adjustments that need to be made.
Okay. Next question. Are these optimization metrics all based on Illumina NGS data, or have you looked at your kit prep optimizations for the ONT platform?
Yeah, so all of these performance metrics and all this stuff that we're talking about here is based on the Illumina platform. We have not used this product, or this product is not compatible with the Nanopore platform. Okay.
Next question. What are the typical allele fractions of artifactual variant calls?
Yeah, so artifactual variant calls usually appear at low allele fractions. And so the publication that I referenced during the talk reports most of these sorts of artifacts appearing below 5% or so. And ultimately what that means is that these types of artifacts become especially problematic when you're trying to detect rare variants.
Okay, thank you. Next, why are chimeric fragments problematic for data analysis?
I guess there's the data quality issues that I discussed during the talk. But in really simple terms, what aligning chimeras during analysis actually is a lot slower, just because of how it has to find, I guess, the position or find the alignments. So minimizing the amount of chimeras ultimately means that you can process data faster.
Thank you. Next question. Can you generate less than 6 KB libraries for long-read sequencing with this kit?
It was not developed to do that, and we haven't done any testing on those at those sorts of lengths. Like I mentioned before, this is really just developed for use on Illumina short read platforms.
All right. Well, thank you, Zane, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Until next time, goodbye.